Continue on with this topic of brain aging, but I'm going to leave a little bit the brain and start going more into the blood and really think about ways that maybe we can target outside of the brain in order to actually treat the brain itself. And the idea behind this is sort of this concept of reversibility. So rather than really sort of delaying a process, can we actually start at a time when there's already some sort of impacts of aging and then reverse it, bring back function. Some people call this rejuvenation. So from the perspective of my lab and the research that we do, we define that idea of rejuvenation from a functional standpoint. So can we improve cognition even within the context of aging prior to disease? And what do I think that that's sort of an exciting approach? Well, we just heard, right, there's all sorts of things that occur with aging, and not everything is declining, but there are some classical types of cognitive function and processes that do decline. A very common one that we often think about is spatial learning and memory. For example, trying to find your car in a parking lot. That idea of just being able to be guided by spatial cues, that declines over age. Now, around the same time that that happens, there's actually an increased vulnerability for neurodegenerative diseases. That tends to happen around the time of retirement age. So sort of in the mid to late 60s, you start noticing a lot of this age-related decline, but you also have an increased susceptibility for neurodegenerative diseases such as Alzheimer's disease. Now, what we're interested in is there's a number of different interventions that are emerging both in preclinical settings as well as just lifestyle interventions that occur in humans themselves. And we know that they can have a profound impact on the aging brain. So what we're really interested in is leveraging these interventions and asking, well, is there some way that we can then target or figure out kind of the underlying mechanisms of these interventions, apply them in the context of aging, and restore function? In essence, increase health span. So again, the number of years that you're going to live may be not necessarily modifiable, but perhaps the quality in which you're living can be. Now, we focus on a lot of different types of interventions, but sort of our preclinical models are really focused on one area of the brain, which is very susceptible to aging as well as neurodegenerative diseases, and it's a master regulator of learning and memory. And that's known as the region as the hippocampus. And here I'm just depicting a cross-section of a mouse brain. The preclinical models that we use are a lot of animal models, rodent-based models, because of the power of molecular biology. And here I'm just showing you in salmon that hippocampal region, and there's a series of cellular hallmarks. So we saw a lot of different hallmarks at the macro level. Here we're going to look specifically within the cellular level. And within that aging component, there isn't gross cell death like there is in neurodegeneration, but there are a number of different changes. And those changes collectively lead to that functional decline. So for example, we have changes in regeneration. Our stem cell pool becomes quiescent. We have changes in neuroinflammation, those resident macrophages known as microglia, as well as the other glia such as astrocytes have a huge effect on that inflammatory process. We have changes at the level of the vasculature, not necessarily just becoming leaky, but actually the type of transport that's going into your brain changes. The selectivity becomes compromised. And ultimately, that's going to lead to changes in the way that your neurons communicate. At the end, neurons, it's just physics, electrical properties. So the way in which those neurons are actually communicating becomes faulty. Those connections are actually less stable. The thing is, each one of these players is still within the brain. So the idea then is, what combination of cells do we actually have to restore in order to bring back function? Do you have to restore all of them to a youthful state? Is it a combination? If we decrease neuroinflammation and restore vasculature, or perhaps it's vasculature and neuronal processes. What's sort of that combination and the minimum amount by which we can actually promote this function? And again, we study a number of interventions that have really been around for a long, long time. Certain ones are lifestyle, like we heard things like physical activity, diet, for example, caloric restriction. Some of them more experimental. For example, we and others have leveraged that model of parabiosis or heterochronic parabiosis, where you sew together a young and an old animal, and you can actually detect a few different things here. You know that there's factors in old blood that are driving aging. We know that there are factors in young blood that can promote rejuvenation. And we know through other models of rejuvenation that you can even dilute out some of the pro-aging factors, and that can rejuvenate. All of this to signal that there's a lot of plasticity left in that aging brain. We've now also gone from things such as parabiosis and interventions now to just young blood itself, or plasma. We can actually reduce it down to that blood product in order to just do simple administrations, and that can actually promote function. And what we're really fascinated by is it that there's individual mechanisms underlying the different interventions? Are there points of convergence? Is it impacting all the different hallmarks of aging, or do different interventions selectively target individual hallmarks of aging? And can those all then have a consequence on cognition? So really what we're trying to do is get down to the cellular and molecular mechanisms. And the reason that we're really interested in this is to the point of actually getting into patients. So we're going and leveraging biology of aging, we're leveraging really basic research for the purpose of identifying molecules that we can then produce in a way that it can actually become a therapeutic. And that's the end goal of the research that my lab is doing. So I'm going to share with you two quick vignettes to give you a flavor for what we think about as blood-based approaches. The first one is just going to focus on this idea of that young plasma itself. Now, we've all, I'm sure, read in the media about, transfusion of blood or injections of blood plasma from younger to older individuals, but that's actually not a tenable approach. You cannot do that lifelong. There's actually not enough plasma, even if we wanted to do it for Alzheimer's patients today, let alone in the next 20 years. So we really need to get down to something that can actually then become a therapeutic. And we believe one way is really to decode those messages. So the first thing that we did is we just took that plasma, we just did mass spectrometry. We just looked at what's changing within the proteins that change with age. And we actually got a signature that kind of pointed us towards platelets, those little cells without nucleus that we think about in coagulation, and also platelet-rich plasma administration for athletes. We then just administered young plasma, a component of that plasma that's enriched for those platelets, and we just looked at the brain, that hippocampal region, and just looked at transcriptional approaches. What's changing in the brain due to these blood administration? And when we looked at the genes that were changing the same after young plasma or that platelet fraction, we saw a lot of things involved in both immune functions, such as chemokines, MCP1, as well as things such as myelination and development. We really focused on those sort of neuroinflammatory components because people had not actually researched whether young plasma could actually have an impact on this. So we, again, zoomed into the hippocampus, now that brain region, and we just looked at inflammatory markers known to change with aging, such as TNF signaling, complement C1q, CD11b, and we saw a reduction in both plasma as well as platelet fraction. Here I'm showing you those microglia, those resident macrophages in red. In that bright green, you see a lysosomal accumulation of a marker, CD68. That's when they're active or reactive. And you see yellow when they're old, and you can see that there's a decrease in that stain when they're infused with these or administered this plasma, again suggesting less neuroinflammation. We then wanted to identify factors, so we then pooled down just that platelet fraction, did proteomic approaches, and now got a number of different factors that are changing, of which one of them that we were really interested in was platelet factor 4. It's also known as CXCL4. And this has actually come across in exercise. It's also come across after neutral blood exchange, where they replace the blood. This is one of the 4 factors that is actually upregulated following multiple of these rejuvenating interventions. So then we just administered recombinant protein this time, and we again looked at inflammatory markers and saw a decrease in TNF, complement, CD11b, as well as IL-1 beta and NF-kappa B. And again, if you look at that sort of green, we have a reduction of that lysosomal marker. Again, less neuroinflammation. Next, what we did is, of course, we wanted to know behavior. Can we recapitulate those cognitive benefits that we and others have seen with young blood by administration of this factor? And here we just did a water maze. So you can see here the little path in red up on the corner. An animal learns to find the little platform, and anytime it goes into the wrong arm, we just call it an error. And the animals get better and better and better at finding the platform. Old animals never quite get as good as a young animal, so they still end up committing 2 to 3 errors by the end of the testing phase. But the old animals that received that recombinant form of that platelet factor were much improved. Again, these were old, naturally old, impaired animals, so we're really reversing that function. We've gone on to do a lot more work since then. This was actually a trifecta of papers that all identified the same platelet factor in the context of exercise, young blood, clotho administration. We've actually gone down and realized that it started decoding the mechanism occurring through the immune system. So we're actually rejuvenating the peripheral immune system. That's having a huge effect on inflammation throughout the body. Now, I'd like to end with this sort of a little bit of what we've been doing with additional interventions. And here we're focusing on exercise. And the reason we focus on exercise is we know that this is really beneficial intervention, even in the context of Alzheimer's disease. The problem, like we heard, is frailty. People can't actually physically do the exercise that we know could be very beneficial. So of course the question that we asked is, well, exercise like parabiosis, like plasma, it's a whole body intervention. Could we just transfer then some of those benefits through blood? And we and others, at Stanford University, Norway, there's a growing body of literature have shown that yes, indeed, you can take the blood of exercised animals and transfer those benefits. So there must be something within that blood. So of course, we really now want to decode that. What are the messages within that blood that are induced by exercise? And through a lot of proteomic analysis, we pinpointed one factor. This is an enzyme known as GPLD1. What's really interesting is this does not change with age. The same levels that you have when you're young, you have when you're old. So it's not a young blood factor, but really it's induced by exercise, even in old animals. And it's actually derived from liver. And what it does is it acts like a scissor. It's just an enzyme that cleaves a little fat tag known as a GPI anchor. Normally what happens is that fat tag tethers proteins to the cell surface. GPLD1 just comes, cleaves it, and then those factors are then cleaved off the cell surface, and they can either go down and activate downstream signaling cascades, they can inhibit downstream signaling cascades, or they themselves can be degraded. So one enzyme can have a huge effect on downstream factors. There's putatively about 150 of them. What we then wanted to know, of course, is can increasing now this liver-derived exercise factor, can that then also recapitulate the benefits of exercise on cognitive function? For this, we use more of a gene therapy approach where we overexpress transiently GPLD1 within the liver, so the liver produced GPLD1 itself. We also expressed a form that was mutated so it could no longer cut. So all of a sudden, we're taking away that cutting ability. And we just asked, what happens to behavior? Now, the effect of exercise is actually really, really robust, and the effect that's recapitulated by this liver enzyme is quite robust, both in the learning and in the testing phase. So you can see in black an old animal control, and in green that old animal that now has increased levels of liver-derived GPLD1. And then when we overexpress the form that can't cleave, the effects go away. So we actually know that it must be acting through downstream mechanisms within sort of like the body itself. Now, ongoing work in the lab right now, done by a really, really talented postdoc, Greg Vieri, is now decoding exactly what's happening. Like I said, there's about 150 putative substrates. They're enriched on the immune system as well as the vasculature. He's doing a lot of really exciting work that's pinpointing vasculature as a really big target. GPLD1 actually doesn't go into the brain, so you can actually target peripheral targets without directly going into the brain and having huge impacts on brain function and brain physiology. So kind of like zooming back out into this big question that I had, can the effects of aging on the brain itself, can that be reversed? We think they can. What's really exciting is we don't think that you have to actually reverse aging across all hallmarks of aging. We think that it can actually be different combinations that can induce plasticity and that can induce that functional recovery. For example, like I said, exercise seems to be really targeting more of the vascular as well as the regenerative or stem cell proportions. The young blood actually seems to have some of the biggest effects on neuroinflammation, less so on some of the other factors. Additionally, we're doing work on things such as caloric restriction. We're also finding evidence that there's humoral factors that can transfer those benefits completely distinct from the ones that we're finding with exercise and young blood. Their sources even being very different. As I said, exercise being more liver as well as muscle, young blood being more immune, and of course, we're still searching to see where the sources of these other factors are. And just zooming back out, really long term in terms of like the therapeutics, can the effects of aging be reversed? We think that they can. We think that we are learning just from normal physiology. We know that our bodies can recover, and we're merely now finding mimetics that we can tap into what your body naturally can do by identifying these individual factors that we're now in the process of making recombinant versions as well as small molecules to try and mimic those effects now and move that forward. With that, I'll just thank my lab, and I don't know if we have any time for questions.
Thank you so much, Saul. That was really a spectacular talk at Tour de Force. Does anybody have any questions for Saul?
Nice studies. Maybe you mentioned this, but if you take blood from older cognitively impaired animals that exercise a lot and treat the animals that are cognitively impaired? Do you see anything then?
Yeah, so the experiment we did was exactly that. So we took old animals, we exercised the old animals and took their blood into the old animals. We wanted to
But the
tease
young was
apart.
more effective, right?
Oh, if you put it back into the young animal.
Yeah.
So we've done something similar, not exactly that. We took animals that were middle age, which means their young blood no longer, it's not young, it doesn't do anything. It basically is a net zero. And we see an additive effect to that. We've never taken the old blood and put it into a young. We've taken middle age, and we've taken young exercisers, and you get an even bigger boost than young blood alone. So there's an additive effect of young and exercise. The exercise can sort of overcome the middle age. We've never done that last experiment, which is the one you want. Yeah.
Okay, do we have more questions for Saul? Fatigue is beginning to set in. Where was that? There was one. Oh, yeah. Do we have a mic there?
Hi, that was really awesome. Can you hear me?
Yeah.
I'm wondering if you guys have ever done an experiment where you've tested a combination of them, so PF4 and GPLD1, I believe, and if those effects are synergistic or if they target the same hallmark and you don't see a synergistic effect?
Yeah, great point. We haven't started doing it on the molecules themselves. We've been playing more with the interventions. Like I mentioned, you exercise a young animal, take their blood. That effect is greater than the effect of just young blood alone. We've been trying to mute some of the old blood effects through caloric restriction. So we've been doing that at the level of the interventions, and the reason why is we actually want to see if there's different factors that get induced by doing those interventions themselves rather than just the molecules themselves, because I think the entire program in your body is changing. A lot of metabolism comes into play, so a lot of what's secreted can be influenced really easily. So we started a little bit broader than that.
All right, more questions for Saul?
Perfect.
Oh, there's one down in the corner. We need to run. It's good for you. It's good for your brain. Yes, getting younger.
But my hearing is still okay. Maybe I can hear
we
you.
need to have the microphone because of the.
Oh, that's right. The
online
recording's
viewers.
online. That's right.
So your platelet fraction also contains these so-called mitovesicles that the platelets release. Have you ever tested those?
So we haven't, but there has been work that has looked at sort of extracellular vesicles, exosomes in the context of young blood and muscle regeneration. We did a little bit of exosome work, but it was predominantly in the brain. And for us, we saw a lot of pro-inflammatory exosomes coming from microglia in the aging brain. We focused less on the inflammatory ones. So for us, they had a big signature in aging. They dynamically changed their content, but that wasn't the blood one. That was all in the brain. Yeah. Great.
All right, thank you so